Okay, Dr. Katz, thank you very much for joining us today. Congratulations on your latest paper that was published this week in the Journal of Neuroscience.
Thank you, Monica.
I was thinking back to the first conversation you and I had and trying to think back when that was. It's a number of years ago. I'm guessing about 8. Does that sound right?
Yes, that's right. It's about 7 or 8 years.
Yeah, and I actually remember it really vividly. I remember I was standing in our family room, and I remember starting off the conversation with, Dr. Katz, you don't know me, but Nino Ramirez suggested that I contact you, and I have a daughter with Rett syndrome, and we had a great conversation. Do you remember that?
I also remember it very vividly. I was sitting where I'm sitting now speaking to you, and you introduced yourself, and you said, we're familiar with your work on BDNF and breathing, and I'm wondering if we can interest you in applying to us for funding. And that, as the more I looked into it, the more I realized that everything we'd been working on might in fact be applicable to Rett syndrome. And since that time, we've shifted the work in the lab so that really everything we're doing now in one way or another is focused on Rett.
Yeah, that's a success story. So I'm really pleased that you became quite interested in Rett syndrome, and now your work may have real clinical relevance. So let's start to talk about the paper. The title is a mouthful. Brain activity mapping in MECP2 mutant mice reveals reversible deficits in forebrain circuits, including key nodes in the default mode network. So our listeners are largely non-scientists. They're parents and family members of children with Rett syndrome. And the main findings of your paper, as I understand it, is that at least in the Rett mice, you found that the front of the brain, the forebrain, exhibits lower activity than normal, while the back of the brainstem, exhibits abnormally high activity. Can you elaborate on this and why you think that that's important?
Yes, Monica. So you have summarized that aspect of the paper correctly. This is really the first time that we've had a map, if you will, a map of which parts of the brain are dysfunctional in a model of Rett syndrome. You know, up until now, there have been studies in a number of labs, including ours, where we look at one particular cell group or one particular circuit, and we see that it's not working properly. But what we wanted to do was generate a global map of what the Rett brain as a whole looks like compared to the normal brain. And so that's the first part of the study, is generating that map. And as you say, we found that there's an entire domain of the brain where activity is reduced compared to normal, and another where it's increased. And so this, in generating this map, we have identified quite a number of brain structures that really have not yet been studied in the context of Rett syndrome. So one of the consequences of generating the map, that this now gives us a very clear picture of where to focus to look at what is not working correctly in the brain in the Rett mice. I think another significance of generating this map is we discovered that many of the key regions where activity is lower compared to normal, these overlap with regions that have also been identified in human patients with classic autism using functional MRI studies in human patients with autism. And we see the same, so in human patients with autism, these areas have also been found to be underactive. And so this now shows a commonality in the pattern of brain circuit dysfunction between the Rett mice and human patients with autism. And I think what this suggests is that there may be common underlying mechanisms contributing to the behavioral symptoms which are in common between Rett syndrome and more classic forms of the autism spectrum. So in terms of the mapping, I think those are the two most significant findings. But I must say, I think there's a third as well. As you well know, as a Rett mother, as the mother of Chelsea, Chelsea has problems with breathing, for example. And that's a behavior which is largely controlled by the brainstem. But her breathing problems get much worse when she is agitated. And they tend to be much worse when she's awake rather than when she's asleep. So how do we understand the interaction between her behavior and her breathing in the context of Rett syndrome? And I think now that we see this imbalance between the forebrain, which is very important for controlling behavior, and the brainstem, which is very important for controlling breathing, I think this helps us understand why we see this interaction between behavior and breathing and in Rett syndrome. Yeah. In other words, the forebrain is not talking properly to the brainstem, if you will. And so I think that is contributing to the behavioral disturbances of breathing in girls with Rett syndrome.
All right. And for the sake of our listeners, the brainstem is a very crucial part of the brain that houses breathing centers, and so it's a very important part of the brain for breathing. You and I have had lots of conversations over the years about how the breathing abnormalities kind of go hand in hand with almost like the state of arousal that the children are in, and that structurally there's really nothing wrong, and their breathing tends to normalize during sleep, which is very encouraging. So how can we make that state of when they're sleeping happen when they're awake, right? What can we learn? So it's complex, but it's really intriguing, and I think your paper starts to answer some of these questions. Can you talk to us a little bit about the outcome measures that you used in the mouse studies? If you could explain them briefly and comment on the significance of these phenotypes to the human condition.
Okay. So in this paper, as you know, in a lot of our work, we do look at breathing and other behaviors, but not in this paper. Here we were looking at, first of all, indicators of brain activity, and secondly, once we saw which areas of the brain, particularly in the forebrain, showed decreased activity, we wanted to assess behaviors that these areas of the brain are involved in controlling. And so we chose something that has a long name. It's called prepulse inhibition of acoustic startle. And I don't think we need to go into the details, but for the purposes of our conversation today, this is a behavior which gives us a window into how circuits in the forebrain are functioning. And thereby, it's a window into cognitive function. And this is also behavior which is known to be disrupted in other neuropsychiatric disorders. And so this test, which we abbreviate with the name PPI, is done the same way in mice that it's also done in human patients. So that was our behavioral outcome in this study. And as I said, I think it gives us a window into how the forebrain, and the cortex in particular, is functioning. And what we saw was that in mice with the MECP2 mutation, that they have an abnormal exaggeration in this behavioral outcome measure. And I'm sure in a moment we'll talk about then how that was reversed with the drug treatment.
So your paper has clinical relevance because you used a drug that's FDA approved and used for anesthesia and for depression and for other disorders, and got a rescue of the deficits that you were seeing. Can you talk a little bit about that?
Yes. So when we saw this abnormally low activity in areas of the forebrain, we wanted to know whether that could be reversed and whether or not reversing it would improve neurologic function in the mice. And so we went into the literature and found that this the drug called ketamine had previously been shown to increase the activity of neurons in many of the areas that we were interested in. And so we gave the drug in a single injection to the mice. And the first thing we found was that indeed it did increase to levels seen in the normal mice. It increased activity in those areas which otherwise had abnormally low activity. And the second thing we found was that drug treatment normalized their PPI response, the behavioral measure that I spoke about a few moments ago. And so not only could it, if you will, normalize the activity map in the forebrain, but it could also normalize at least this one aspect of neurologic function. We've gone on now. We've obviously we're pursuing these studies and looking at other aspects of neurologic function. But the excitement here is that, as you say, this is an FDA-approved drug. However, it is also a potentially very dangerous drug. And so we do not recommend that anyone goes out and starts using it at this time. It needs to be studied very carefully in individuals with Rett syndrome to see if it is safe in that context.
It is amazing how fast it works, though, isn't it?
Yes.
In its use in depression, patients say that they feel better sometimes within hours.
Yes.
But yes, it does. It can cause psychosis and hallucinations, and especially in a nonverbal population, that could be concerning.
Right, because they would not be able to tell you if they were experiencing those potential side effects. So what's interesting in our studies and also in the work being done in depression is that we're seeing these positive effects in the Rett mice at doses that are much lower than produce those concerning neuropsychiatric effects. Mm-hmm. And it's doses that do not produce anesthesia as well. And as you said, both in patients with depression and in the Rett mice, we see very rapid effects within an hour or so. And that's because the drug actually modulates the electrical activity of the nerve cells. And that's something whose effect can be seen quite quickly.
I mean, this is a fairly old drug. It's been around about 50 years.
Yeah.
But you know, even this week in Science, the issue that came out today, actually, there's a review article on the use of ketamine for depression, and there's a whole lot of excitement around it now. So it's a drug that seems to have found a new life, so to speak.
Yes, it's correct that it is experiencing a renaissance because of these findings in depression. And I must say that I think the Rett field is really going to benefit from the work on ketamine in depression because of the findings in depression. There are a lot of people working on understanding in more detail the mechanism of action of ketamine. And we believe that there may well be safer drugs available that have similar actions as ketamine or that work on some of the same mechanisms that ketamine works on. And so as people are looking at this in more and more detail, we will benefit from their findings in the Rett field.
Yeah, I actually recently met one of the two scientists who actually did the initial work linking ketamine with the relief of depression. His name is John Crystal. He's chairman of the Department of Psychiatry at Yale. And I will certainly send your paper to him and be interested to hear his comments. I know there's also work going on trying to design drugs that can mimic the benefits of ketamine, but hopefully not the side effects. So that's also an area that hopefully the Rett field can benefit from.
Yes, and this actually dovetails with the reason you called me 8 years ago, and that is our work on BDNF. One of the things that ketamine has been shown to do is to increase levels of BDNF, which, as we and others have shown, this protein called BDNF is in deficit in the brain of Rett mice. And so that may be one of the mechanisms by which ketamine is having its effects. And so we may, as you know, we have other projects in my lab that are looking at enhancing BDNF signaling as a therapeutic strategy in Rett. And so there may be overlap with the ketamine effects and those mechanisms as well.
So since your paper came out, which is just a day or two, but I received a number of emails from parents who saw the press release and who told me that they saw benefits in their daughters after procedures where anesthesia was used. Maybe dental procedures or maybe some surgeries, and now they're interested in going back and seeing if they can find out through the doctors who did the procedures to see what was used. It would be really kind of interesting to know that. And the benefits that they saw lasted 2 to 3 days and were a much increased attention span and alertness and eagerness to communicate, which then went away. Now, other people have, there's been some discussion on the Facebook pages about this, and other parents are saying, no, we see the opposite. It took weeks for our children to, for the anesthesia to wear off, and anesthesia can be problematic in Rett syndrome children. But it's interesting to hear from some cases where the opposite happens and the children are really, really alert. And I'm wondering whether we might learn something from these cases.
Yes, well, I'm really intrigued by what you're saying because I've been looking for those kinds of reports. I've not found anything in the scientific literature. But it is true that ketamine is used for some surgical procedures in children. It has been used for anesthesia in children with Rett syndrome. And so I'd be very interested actually in speaking with the parents you're referring to once they get information on what anesthetic was used. Ketamine would not be used in all circumstances, so maybe some children had it and some didn't, and that might account for the different reports that you're hearing. But in the depression literature, a single infusion of ketamine produces antidepressant effects which can last for up to a week. Mm-hmm. So if these girls indeed did receive ketamine, it would not surprise me that they had effects lasting a few days. And what you describe, alertness, attention, and so on, these are exactly the areas in which I would expect to see an effect. But again, at this point, it's just speculation. But there may be some information out there to be gleaned from parent reports.
Absolutely, and I think so. For any parents that are listening whose children had benefits, please get in contact with me because this could be an area where anecdotal parent information can help inform the scientific process. So what are your next steps with regards to this work?
Right. Well, we're moving in a number of directions. One is that we are speaking with Rett clinicians and psychiatrists who do clinical trials for neuropsychiatric disorders in both adults and children. So we are beginning to look at how we could translate these findings into preliminary clinical studies. Of course, the first step there will be to establish safety in the patients, in individuals with Rett syndrome. Secondly, we're looking more broadly at just how many areas of behavior are improved in the mice with ketamine treatment. A third area is what we were speaking about earlier, is to understand exactly what is the mechanism of action by which ketamine is reversing symptoms in the Rett mice. Because if we can identify the specific mechanism, it's possible that there will be even more specific drugs that are more specific and therefore might have fewer side effects than ketamine. So these are all the areas in which we are moving ahead.
Do you have any plans to look at breathing with plasmography technology? Or
Yes.
Yeah.
Yes, definitely. We actually are just starting those experiments.
Okay. It'll be interesting.
Now, again, as we're learning, I think all of us who are doing this kind of work, we know that there are many systems in the brain which are not working properly as a result of the MECP2 mutation. And so we don't really expect that there will be necessarily any one drug that fixes everything.
Yeah.
And so what we're interested now in finding out is exactly what areas of behavior and neurologic function are improved by ketamine, and we also want to know which ones are not. Because ultimately we believe that it will be a cocktail of drugs, if you will, or a combination therapy that will provide the biggest benefit.
Yeah, I mean, I think from a parent's perspective, I think we have to keep in mind if they're thinking about, the dramatic outcome of the reversal experiments that Adrian Bird did, that was because he got to the root of the problem and turned on MECP2, and all of the downstream effects from that just kind of naturally fell into place. And so to achieve that kind of dramatic outcome, I think you really need to go to the root of the problem, and that might be through gene therapy or activating the silent MECP2. But FDA-approved drugs that may act on a subset of symptoms, like you said, is not going to be it's not going to cure all symptoms. It'll achieve, hopefully, a subset of them. But, I think parents would be happy to have any of the multitude of symptoms improved.
Right. I think we don't know when the day will come that we'll have the technology to fix the gene defect.
Yeah.
So in the meantime, what the strategy in our lab and many others is to try and identify what are the key functions that are disrupted by the gene mutation and making them targets for drug therapy.
Absolutely. I mean, these are all important approaches, and it's important for us to have the resources to pursue all of them in parallel.
Right.
So.
Because as I know you appreciate deeply, if we do find FDA-approved drugs that can reverse even some of the symptoms, then the pathway to clinical trials is much, much shorter.
Yeah.
Because we already have a lot of clinical experience with these drugs.
Absolutely.
So that's the hope.
Is there anything you'd like to add, something that I didn't ask, or anything that you'd like to have the Rett lay community know?
Well, I would conclude by saying that the genetic reversal experiments by Adrian Bird that you mentioned were really a paradigm shift for the entire field, because he showed that, of course he did something in mice which we cannot yet do in humans, but he showed that if you could restore MECP2 function, you could recover a tremendous amount of neurologic function. You could reverse symptoms. So then the question became, can we do that with drugs, since we don't have the technology to do it genetically? And so I think what our study does is provides new evidence that indeed using a drug strategy rather than a genetic strategy, we still see that we are able to reverse symptoms in mice which are already sick. And so I think that just encourages us that the possibility for reversibility, at least in the mice, is real, and it's something, by real I mean that it's something that can be affected using drugs. And so this gives us more ideas about what drugs might in fact be effective in girls with Rett syndrome.
Yeah, I agree wholeheartedly with that comment. I think your paper adds to the portfolio of data that we should remain optimistic, and that the Rett brain, at least in a mouse, is malleable, and that there's no irreversible damage. and that we all have to continue to keep working on trying to figure out how to rectify those deficits. So thank you so much, Dr. Katz, for your work and your continued interest in Rett syndrome, and we look forward to hearing more news from your lab.
Thank you, Monica. Thank you for your continued interest and support for our lab. I think I also want to end the interview with the note of optimism that you just struck. I think this is a very hopeful time.
Thank you very, very much.
Thank you, Monica. Bye-bye.